Oh, hello students. Today we will just learn some questions and answers which can be used for admission test in BS programs. So the first question that can be asked is, what is DNA? So DNA is genetic material in all living organisms that carries instructions for growth, development, and functioning. So it's abbreviation of deoxyribonucleic acids. So then we come to this. What are the differences between DNA and RNA? So DNA is double-stranded, contains thymine and stores and genetic information. RNA is single-stranded and has uracil instead of thymine and helps in protein synthesis. So what are the differences between DNA and RNA? Our DNA is double-stranded, contains thymine and stores genetic information. RNA is single-stranded and has uracil instead of thymine in addition to adenine, guanine, and cytosine and helps in protein synthesis. So we can see that another question can be, what are enzymes? Enzymes are biological catalysts made up of mostly proteins that speed up chemical reactions in living organisms. So the proteins which speed up or catalyze chemical reactions will be termed as enzymes. And what is the nature of enzymes? Mostly they are protein in nature, but they can be RNA. What is fermentation? Fermentation is a metabolic process where microorganisms convert sugars into alcohol or acid, often without oxygen. So fermentation is the metabolic process where microorganisms convert sugar into alcohol and carbon dioxide or acids, often without oxygen. So define genetic engineering. So genetic engineering is the process of modifying an organism's DNA to introduce new traits or functions. What is recombinant DNA? The DNA which is made by combining two DNAs from different organisms and to make a recombinant DNA molecule. By combining two more DNAs from different organisms and ligating them. What is PCR? So PCR is the polymerase chain reaction, which was invented by Carey Mullis in 1983, and it is used for the synthesis of DNA or to make multiple copies of small segments of DNA. What are stem cells? The stem cells are undifferentiated cells that can develop into different types of specialized cells in the body. Do you think zygote is a stem cell? Yes, it can be termed as a pluripotent stem cell because it has this power to develop in a complete animal or plant. So what is the central dogma of molecular biology? So central dogma of molecular biology, it explains the flow of genetic information from DNA to RNA and from RNA to protein. So through the process of transcription, this information stored in the DNA will be transferred to the RNA, and with the help of translation, this protein synthesis will take place, which will be based on the message in the messenger RNA, and the messenger RNA message will be based on the DNA. So the flow of information from DNA to RNA and from RNA to protein is termed a central dogma of molecular biology. So what is a plasmid? Plasmid is a small, double-stranded, circular, self-replicating extrachromosomal DNA in the bacteria and in some yeast is termed as plasmid. So plasmid is a small, circular, double-stranded DNA molecules which are self-replicating and they are extrachromosomal. So they can be used as a vectors because they can be extracted and they can be restricted with the restriction enzymes, and then we can add and ligate segments of DNA, and then we can clone them, and then we can transform them. So what is transformation? So transformation is the process of uptake of DNA by a cell from its environment. So, and this process was discovered in 1928 by Frederick Griffith. So what are restriction enzymes? So restriction enzymes are proteins in the DNA, and that proteins that cut DNA to specific sequences used in genetic engineering. So restriction enzymes are the proteins or enzymes that cut DNA at specific sequences used in genetic engineering. And what is biotechnology? In simple words, biotechnology is the use of living organisms or their parts to make useful products for the humans. Mostly use of bacteria and microbes for the benefit of human beings will be termed as biotechnology. So give an example of a genetically modified organism. So Bt cotton is a genetically modified organism that produces its own insecticide to protect against pests. What is cloning? Cloning is the process of creating genetically identical copies of an organism or cell or DNA. What is the difference between mitosis and meiosis? Mitosis makes the identical cells for growth in somatic parts, and this meiosis is the process which takes place in the reproductive organs to produce germ cells or gametes, sperms, and eggs. What is the function of ribosomes? Ribosomes are the sites in cells where proteins are synthesized. What are antibiotics and how they do work? Antibiotics are drugs that kill or inhibit the growth of bacteria by targeting their cell walls or metabolism. So antibiotics are the drugs that kill or inhibit the growth of bacteria. So what is CRISPR technology? CRISPR is a powerful gene editing tool that allows scientists to precisely modify DNA in an organism. How does vaccination work? Vaccines stimulate the immune system to recognize and fight pathogens by introducing harmless forms of the germ. What is an antigen? An antigen is a molecule that triggers an immune response, often a protein on the surface of pathogen. What's the difference between a prokaryotic and eukaryotic cell? Prokaryotic cells lack a defined nucleus and organelles. Eukaryotic cells have both defined nucleus and cell organelles. What is photosynthesis? So photosynthesis is the process where green plants use sunlight to make food from carbon dioxide and water. What is the genome? A genome is the complete set of DNA, including all genes in an organism. What is the gene expression? Gene expression is the process where information from a gene is used to make a functional product like a protein. What is microbial biotechnology? It involves using microorganisms like bacteria and fungi to produce useful products like enzymes, antibiotics, and biofuels. So what is bioinformatics? So bioinformatics is use of computers and softwares to analyze and interpret biological data, especially DNA sequences. Would you like to proceed? So, yes. So, what is RNA? RNA is ribonucleic acid, is a nucleic acid involved in protein synthesis and gene expression. What are the components of DNA? DNA is made up of nucleotides containing a deoxyribose sugar, phosphate group, and nitrogenous bases. So, this, what is made up of? DNA is made up of nucleotides. And what is the nucleotide? Nucleotide is the units of this DNA structure, and the major components of the nucleotides are nucleotide is made up of a deoxyribose sugar and phosphate group and nitrogenous base. And this nitrogen base can be A, T, or G, or C. So what are the components of RNA? RNA is made up of nucleotides with ribose sugar, phosphate group, and base. And these bases are adenine, uracil, guanine, cytosine. What are the nitrogenous bases in DNA? Adenine, thymine, guanine, and cytosine. What are the nitrogenous bases in RNA? Adenine, uracil, guanine, and cytosine. What is the structural difference between RNA and DNA? DNA is double-stranded and RNA is single-stranded. What sugar is present in DNA? Deoxyribose. What sugar is present in RNA? Ribose. And where is DNA found in a cell? In the nucleus and mitochondria in eukaryotes. And where is RNA found in a cell? Where is RNA found in a cell? So in the RNA is found in the nucleus and cytoplasm. What's the role of DNA? Store and transmit genetic information. And RNA, what is the role? It helps in protein synthesis. And there are three types of RNA: ribosomal RNA, messenger RNA, and transfer RNA. A messenger RNA will take the message from DNA to the ribosomes, and transfer RNA will bring required these amino acids at the site of protein synthesis, at the site of ribosomes. So this, what is a gene? A segment of DNA that codes for a protein. And what is the central dogma of molecular biology? Flow of information from DNA to RNA to the protein. And what is messenger RNA? Messenger RNA that carries genetic information from DNA to ribosomes. And what is tRNA? Transfer RNA brings amino acids to ribosomes during protein synthesis. And what is DNA replication? The process of making a copy of DNA. What is transcription? The process of copying DNA into RNA. What is translation? The process of making protein using ribosomes and tRNA and messenger RNA. The process of making protein using messenger RNA, transfer RNA, and ribosome is termed as translation. What is complementary base pairing in DNA? Adenine pairs with thymine and G pairs with C, guanine with cytosine. What is the complementary base pairing in RNA? This adenine will pair with uracil and guanine with cytosine. And what enzyme is involved in DNA replication? DNA polymerase is involved. And what enzymes are involved in RNA synthesis? RNA polymerase. What is a codon? A sequence of three RNA bases that code for an amino acid. What is a mutation? A change in the DNA sequence. A change in the nucleotide sequence of DNA will be termed as a mutation. Can RNA be reverse transcribed into DNA? Yes, with the help of reverse transcriptase, we can convert RNA into DNA. And who discovered the structure of DNA? So Crick and Watson in 1953 reported the double-stranded DNA structure. And what is RNA? Why is RNA less stable than DNA? Because it has a hydroxyl group on the carbon number 2 of ribose sugar. So RNA is less stable than DNA because it has a hydroxyl group on the carbon number 2 of ribose sugar. Okay, thank you.